Okay. Okay. Hi, everyone. Thank you for joining. Am I audible? And is my screen visible? Are you able to see my screen? Okay. Okay. Thank you so much. Thank you for the confirmation. Okay. So, welcome to problem solving session 9. And as the image on my screen already depicts of what we are going to discuss today, we are going to mainly talk about photosynthesis. Prior to this, we have had two sessions where we covered about how we are dealing with Krebs cycle and glycolysis, gluconeogenesis. And today we're going to see, so we saw how ATP synthesis takes place for oxidative phosphorylation. Today we will see about photophosphorylation, which is photosynthesis. Okay. And as you would have seen in the notes that it covered two aspects, light reactions and dark reactions. So these are the two parts that we'll be covering in today's session. Right. So coming to photophosphorylation is basically photosynthesis. So what is photosynthesis? It's actually, as we most commonly know this as being present in plants, but we also know now that it's present in variety of bacteria and unicellular eukaryotes like algae. So what happens in this organism, or what happens in this process is there is a capture of solar energy, uh, which are by the photosynthetic organisms, and that is converted into a chemical energy of reduced organic compounds. And this reduced organic compound is the ultimate source of nearby all biological energy that we see in these organisms. Right now, photosynthetic organism, they trap the solar energy and then they form ATP. That is what anyway in previously also we have seen. But previously, in terms of in the oxidative phosphorylation, we have seen that the currencies that were being generated were NADH. However, in the photophosphorylation or photosynthesis, along with ATP, these organisms form NADPH. This is the difference that you need to remember. So in the previous one, we saw how NADH is formed. In this, we'll see how NADPH is formed, right? And then the organisms, they use the ATP and the NADPH as their energy sources to make carbohydrates, which is carbon assimilation, which is the dark reactions that we will see. Okay, and along these are the so they have carbohydrates. Along with this, they also form several organic compounds using carbon dioxide and hydrogen water. So we'll see, as you can also see in the image on the slide here, that's carbohydrate that is formed and that's water that's being used, right? So you have usage of water and carbon dioxide to form carbohydrate using, and the energy currency, the energy ATP and NADPH, right? Now these photosynthetic organisms, as you can see on the image here, so these are using this photosynthetic cells are using solar energy as their source of energy, like the ultimate source of energy. And then there is a cycling of, let me just start the pointer just a second. Yeah. So you have the solar energy that has been taken by the photosynthetic cells, and they are utilizing oxygen and carbohydrates. Of basically, they are giving out oxygen and carbohydrate, which are used by the heterotrophic cells. These heterotrophic cells give out carbon dioxide and water, and these are used by photosynthetic cells. So there is a balance between the photosynthetic cells and the heterotrophic cells in the nature, and because of their products that they generate and use up, right? So this is, and if you go into the details of what happens exactly in photosynthetic cells, so this is what happens. There are two parts of photosynthesis: light reactions and carbon assimilation. Light reaction builds up the energy currency. So what, by how? By splitting the water. So the solar energy is actually used in splitting of water, which is given by something called Hill's reaction. So you can see that you have a splitting of water by driving the energy, and in the meanwhile, we'll see how electron is getting transferred, and ultimately you have the formation of NADPH. right? Now this NADPH is formed, and in the next part you have the use of this NADPH and ATP, which will be used in reactions called carbon assimilation reaction to finally use up the carbon dioxide to give carbohydrate. So these are the overall reactions that you see. So water splitting, H2O and NADP giving you the NADPH, which is the product of the light reaction. And then you use up the water and carbon dioxide to give you carbohydrates. So we will see how these steps are taking place. Right. Now, photosynthesis in plants, where does it take place? We know about mitochondria when we read about Krebs cycle, we read about TCA. Then we also saw about oxidative phosphorylation. We saw the structure of mitochondria and why was that important? Because we have the inner mitochondrial membrane and the outer mitochondrial membrane and the intermembrane space. This was important for us to remember which was the N side and which was the P side, which was important to understand the chemical gradient that was formed, chemiosmotic gradient. So we looked into the structure of the organelle where this was taking place. So similarly here, for photosynthesis, the site is chloroplast, right? And this chloroplast have both the light dependent and carbon assimilation reaction. Now, if you look into the structure of the chloroplast, you will see it is very similar to mitochondria. You will see an inner membrane, this thin lining here. If you go towards its right, the third number marked here is the outer membrane. The second number marked is a membrane space between these. So this is the intermembrane space. The fourth, which you see here, is a stroma. Stroma is basically what is being lined by the inner membrane. And then you have these two very important constituent. Fifth one is called thylakoid, and sixth is the lamellae. Okay. Now, thylakoid is basically stack of membranes. So these thylakoid membranes are basically stacked together in a grana. So all these are vertical stacks of thylakoids, and these are called grana. So you have these grana, and the membrane of these grana is called the lamellae. And we will see why is it important, because this is the side where you have the capture of solar energy, right? So in the thylakoid membranes, which is the lamella, there are photosynthetic pigments. Photosynthetic pigments are basically the ones that are involved in photosynthesis. So photosynthetic pigments and the enzyme complexes that carry out these reactions. So where does the difference actually take place between light reaction and dark reaction? In the site. So light reaction takes place in the lamella or the grana here, while carbon assimilation reaction takes place in the stroma here. So in the stroma, which is the outside space, there you have the carbon assimilation reactions, while the process of capturing of solar energy and making use of it to generate ATP and NADPH is taking place in this grana, right? Now, so we have used some terms like photosynthetic pigments. What do these terms mean? These terms basically mean that they are pigments, or they are compounds which are able to absorb light, right? And photo means light, pigments mean which are able to absorb the light. Now, what do they do when these components absorb light? They get excited. So when a molecule that has absorbed a photon gets excited, it's called an excited state. This excited state is generally unstable, so it has to come back to its stable form. So what happens concomitantly or along with this is an electron which is lifted to a higher energy orbital. This usually returns rapidly to a normal lower energy orbital, and this is what is called the decay of the excited molecule. So the excited molecule is basically an electron that has shifted to a higher orbital. It has come back to its ground state, or it has to come back to a lower energy orbital. So these pigments, they are absorbing light, getting excited, and then they're coming back to ground state, right? Now, while doing this, what is the type of decay that can happen? This can simply be like the shift of the electron to the next orbital, or what happens here is that there is a transfer of excitation energy from the excited molecule to a neighboring molecule, Just as the photon is a quantum of light energy, so this exciton, or this whatever this pigment that has got excited, is a quantum of energy which is passed on from one excited molecule to another. We all will become clear as soon as we start moving into the components and see how it makes sense. But what you now need to remember is that what is driving this entire electron flow here is the excitation of these pigments. They are taking solar energy, they are absorbing photons, making use of the energy and getting excited, and while coming to the ground state, they're transferring these electrons into the ETC, or the electron transport chain, right? Now, who are doing this? What is present in the thylakoid membrane that's making it do all of these absorption? And we know the term, obviously, and it's already on the slide that you can see. It's chlorophyll. Chlorophyll, we have already heard these terms, I think, since childhood. So chlorophyll is a light absorbing pigment in the thylakoid membrane. And green, they are green colored pigments because they reflect green color. And it's not the only light absorbing pigment that is there in the plants, or that is there in the chloroplast. You have several other components, and all these pigments are there on your slide. You have phycocyanin, lutein, beta carotene, and all these are important because they absorb light at different they have different absorption spectra. What does it mean? At what wavelength of light will they get excited? It's different for different components, and it's important because you need, or think of it this way, you need to maximize your energy gain. So if you have many such chlorophylls which have different phytopigments or photopigments that can capture light at different wavelength, you're basically capitalizing on it, that you can ensure maximum energy retention, right? Now, so you need to remember chlorophyll. You need to remember there are two types of chlorophyll, chlorophyll A and chlorophyll B. Now, chlorophyll is not, like I said, chlorophyll is not the only protein. Chlorophyll is present along with specific binding protein that forms the light harvesting complex. The name tells the function. They are light harvesting complexes. Basically, they are all functioning to capture light from the sunlight or from the solar radiation, right? And so the graph here is basically telling you about the absorption. We know the visible light is between 400 to 700 nanometer, and this is a spectrum for the absorption of all the photopigments which are capable of absorbing visible light. And as I told you, plants are green because this component here, chlorophyll, as you can see, it reflects green color, or it transmits green color, which is why we perceive the plants or leaves as green color, right? Then there are several other components that give off different, that absorb at different wavelength and give away the reflectance at a different wavelength. But what you need to remember is all these different components are present as a light harvesting complex to maximize their uh, intake or maximize their absorption of light energy, right? Okay, just a second. Yeah. Now, coming back to it. So the light absorbing pigments of thylakoid or bacterial membrane, like I mentioned, these processes are also there in bacterial membranes. We will see the difference between the processes taking place in thylakoids and bacterial membrane. What is the difference between these two processes? But these pigments which are present in both the membranes, they are arranged in functional arrays, which is called photosystems, right? Now, all the pigments molecule in a photosystem can absorb photon, right? So everybody is absorbing photon, but you only have certain chlorophyll molecules which are associated with a photochemical reaction center that specialize in transducing light into chemical energy. So let's break it up and understand. You have a chlorophyll and you have several free chlorophyll. This chlorophyll, the central one, let's say, is bound to several other components that we will see in a while. So this one is actually able to take energy from solar radiation and then transmit the energy to other electron carriers, right? But the other chlorophylls are free. So if they are free, then what's the point? How are they taking part in photosynthesis? What happens in those free chlorophylls is they take the light energy, they are able to do that, but they don't convert it into chemical energy. They help rather in exciting the next chlorophyll. So the free chlorophylls that you see, they all transmit their energy to this one reaction center, right? So that's what is mentioned here. So all the pigments that you see, all the pigments that we have just seen in the previous slide, they are all capable of absorbing light, but only the few chlorophyll molecules that are associated with the photochemical reaction center, they are specializing in using this energy. While the other pigments are, as I call, they are called antenna molecules because antenna, as you know, by the term, they are used to capture signals. So they are in this sense, they are used to capture the sunlight. They also absorb light energy and then transmit it to the reaction center. So on this slide, you can see this is the reaction center. So you have the photochemical reaction center here, and this is where the conversion of light energy into chemical energy takes place. And along with this, you have the chemiosmotic gradient. While the other molecules that you see, these absorb light energy and then transmit it to the reaction center. So your light harvesting molecule or the light harvesting complex just doesn't have one chlorophyll molecule, right? Okay. Now this is very important. This is what you need to remember. So on this slide, you have two photosystems. Photosystems are basically from the start till the end, how does your electron transmit upon getting excited by your solar radiation, right? So you have two type of such systems. One is in purple bacteria, or that is also called the pheophytin quinone type, and the other in green sulfur bacteria, which is the FES type. These are not difficult concepts to understand. What you need to remember here is how they are structured. What are the components in chain? Which components follow the other? That is what you need to remember. Okay, right. So let's look at the first. So first let's try to understand what's happening. So the central component of the first um, reaction center, first type of photosystem is pheophytin. Whereas here in type 1, you have quinone and a ferredoxin. That's the first difference that you need to remember. That in one, you have pheophytin, which is the first electron acceptor, while in the type 1, you have quinone and ferredoxin, right? This is type 1, this is type 2. Don't get confused, okay? In type 2, after pheophytin, you have quinone, then the cytochrome BC complex, then cytochrome C. This is the cycling. So the electron is transferred back to the original state. While here you do have cycling, but you also have some loss in the form of ferredoxin. Okay. This minute things is what you need to remember in terms of photosystems 1 and 2. Right. So let's look into this a bit more detail. So in the pheophytin quinone reaction, or the type 2, or the first one that you see here, so the photosynthetic machinery here is that a single reaction center, and the reaction center here absorbs the wavelength at 870. So a single reaction center, which is at 870, that along with cytochrome BC1 complex is present, right? Now here, what you see is that the illumination, upon illumination is basically when you have solar radiation. So upon illumination, this drives electrons through pheophytin and a quinone to cytochrome BC, and then after passing through the complex, electron flow through cytochrome C2 back to the reaction center. So that's the cyclic flow. Okay. Now, if you see in the purple bacteria, uh, there is a cycling of the electron that is being generated. But in the first one, or the type 2, which is type 1, which is shown in the second part here, that is in the green sulfur bacteria. This has two routes for electron transfer. And the excitation wavelength is also different. It's 840. The reaction center has an excitation at 840. So there is a cyclic route that passes through quinone and cytochrome BC complex, and then back to the reaction center via cytochrome C553. But there is also a non-cyclic route from the reaction center, which is via this FES protein ferredoxin, and that is what is leading to the formation of NADH. Okay, so this you need to remember that the type 1 has both cyclic and acyclic. The one that is non-cyclic is giving rise to the NADH. Okay. Now this was about bacteria. Purple bacteria have one type cyclic. The green sulfur bacteria don't have cyclic. Here you will not see one thing, which is basically the splitting of water. So you will later see that in these, the compounds which are used to drive energy are different than water. But when you go to plants, sorry, yeah. When you go to plants, you will see the first step is basically a oxygen evolving complex. This is the step where they give, or where they are giving out oxygen. They are basically using the energy from solar radiation, or via illumination. They're splitting water to give oxygen. So in plants, if you see, the same two systems that we have just looked into, they both are present. So you can see photosystem 2 with a pheophytin and photosystem 1 with the ferredoxin. Both of these are present. So what exactly happens in plants then? So if you look into plants, basically you have two of these reaction centers acting in tandem. And if you look into the structure, or just visualize this flow of electrons, you see something like this. One direction, two direction, three direction. And what is this forming? This is forming a Z. So generally, the electron transfer in a plant is called as a Z scheme, or Z flow of electrons, Z type electron flow. So Z scheme is basically the photosystems, in the photosystem, how the electron is transferring. So you need to remember these terms as well. Right. Now, the Z scheme here showing the pathway of electron transfer from the water, which is the first reaction that takes place, to NADP, which is the last electron acceptor. Right. This is, however, in a non-cyclic photosynthesis manner. Right. Now, the what happens here is, to raise the energy of electrons which are derived from water to the energy level that is required to NADPH. As you can see, the electron energy difference is there. Each electron must be lifted twice by photons, and we will see these stoichiometry as equation in a while. But just to understand, so to drive this reaction, each electron is lifted twice by photons, PS2 and PS1. The one of the photon is required per electron in each photosystem. So each photosystem makes use of one photon each to lift one electron, right? Then after this lifting, after this excitation, these high energy electrons flow downhill. As you can see, they have raised. Now they are coming back. So while they are coming back, they have raised. And while they are coming back, they are coming to these reaction centers of these coming to these compounds which have low, which have the tendency of accepting electrons. So they are moving from downhill via an energy gradient through the carrier chains, right? Now protons, as we have known so far, that whenever there is this electron flow, there is proton that is being pumped out from the membrane. Here also, the protons move from the thylakoid membrane. So it moves across the thylakoid membrane during this water splitting reaction. And this electron transfer and proton movement take place together. This proton transfer gives rise to something called proton gradient that we already know. And that proton gradient is then further used to generate the ATPs. Right? So that's your system in plant. Now, as I said, that we look into the stoichiometry of the usage of the reaction. So we see that overall in the plants, you have two water molecule that is being split to give one oxygen molecule. In the meanwhile, you have two NADP that has been reduced to NADPH, and two protons also have been given out, which is moving out. And in the meanwhile, as we know, to do this, they are using 8 photons. 4 photons are being used by the first photosystem. Second photon, or the remaining 4, is being used by the photosystem 2. Right? So what you need to remember, the cue for you to, when you are doing this revision, look into the equations, look into the stoichiometry, because to understand conceptually, it's very easy and it's not that difficult. But you need to remember, and we will do questions on this also, that you have to look into the stoichiometry and the ultimate equation and how many molecules are being used and generated. Right. So now coming to this is what I was talking about when we were talking about light harvesting molecules. So as you see here, that this is basically a schematic drawing of the essential proteins and cofactors in a single unit of photosystem 1. There is a large number of antenna chlorophylls that you see here, and that surrounds the reaction center. And when these get excited by illumination, they are conveying this energy. This is, as you can see here. So let's say these are the antenna molecules that are being getting excited. So they get excited and then transferring this to this chlorophyll, which is the main reaction center. Right. So this is the basically the pictorial representation of how this actually takes place. Okay. Right. Now, coming to the ATP synthesis, which is the ultimate reaction that takes place during your photosynthesis. Now, this is very simple. This is, if we have understood the ATP generation in the oxidative phosphorylation, this is also easy to understand in terms of photosynthesis. So here, as you know, as you can see here also, let me show you. So you have all these PS2, PS1, all this song and dance happening, and these are on the membranes. And you have, remember, your protons are being moved across the membranes. The protons are moved here into the lumen, and that is called the P side. So you have the protons here. Now here only on the membrane, you have the ATP synthase, and these protons, because they are more on the lumens, they can move across their concentration gradient, and that is via the ATP synthase F naught component or subunit. And in the meanwhile, when they are moving across this subunit, they are also generating ATP. How do they do this? We have already looked into the mechanism. There we have seen something called rotational catalysis, where we saw how each H plus that is entering is rotating the ATP synthase, and that's giving rise to ATP. So that we have already done in the previous session. So if you have any doubt, you can look back into those sessions again. But what we need to remember here is similar, absolutely similar process is taking place here. Even in photosynthesis, you have the electrons, which are depicted by the blue color here. So you have the electrons that move from the H2O through PS2, through the intermediate chain of carriers, then through PS1. Remember, PS2 is first and then PS1. PS is basically photosystem. So photosystem 2 to photosystem 1 and finally to NADP, that you can see the entire chain you can see there. Now, we have just realized that they use two photons. This is one photon. This is the other photon. This is the water splitting. This is the molecule generation. And this is how you have the H plus, which is being transported out into the lumen. Right, and this is the flow of electron which finally reaches to the PS1 and then ferredoxin, and then NADP is getting reduced. Right, the protons that are pumped into the thylakoid lumen, as you have seen, by the flow of electrons, re-enter the stroma. So this is from the stroma to the lumen, so they're coming back to the stroma, and this happens via ATP synthase, and we know the mechanisms already, and that leads to ATP generation. Now, if I've already drawn parallels to mitochondria multiple times during our conversation here right now, and that kinds of already gives us an idea that the topology or the type of proton movement or the way of moving of proton and the way the ATP synthase is oriented is similar in the membranes of mitochondria, chloroplast, and in terms of bacteria, where also you have these photosynthesis taking place. Right, now, as you can see here, this is mitochondria that we have already discussed in the previous session. So you have from N side protons into the P side. Let's first just look into the movement of protons. So in mitochondria, you have the matrix N side and intermembrane space as the P side. Protons is moving from N side to P side. If you see chloroplast, you have stroma as the N side and thylakoid lumen as the P side. Again, H plus or protons is moving from the N to P. In bacteria, cytosol is the N side and intermembrane space is P side, and the movement of protons is from N to P. So this is perfectly sitting for all the three systems. Now let's look into the next part, which is basically how these protons, which is in excess in the p side, how are they taken back? They are taken back because we know that these membranes are not permeable. So they are taken by specific transporter, which is the ATP synthase. And in all of these three intake, you can see the part that is taking place is FO or F naught. That is the part that is taking place in taking the H plus inside, while concomitantly you have the ATP generation. So all of these are basically same reaction mechanism that is being taking place, right? And what does this tell us about you know um, homology? It basically tells us that all these genes are basically same. They all are same. That's why they all are coding for similar kind of proteins, and they are all homologous to each other. What is all homologous to each other? Not only ATP synthase, but also cytochrome B6 complex, the main major components of photosynthesis or oxidative phosphorylation. You will see that they are all functioning in similar ways. They are all homologous to each other, right? So this should give you an idea that the reactions that we see or the pathways that we see in different biological systems, they are highly conserved, right? They are being performed by similar kind or homologous proteins, right? Now, including all the ADP and ATP transformations or formation, this previous equation which we had discussed here can be modified into this overall reaction. Same, two water molecules giving one oxygen molecule. 8 photons are being used to reduce 2 NADPH, which is giving, sorry, 2 NADP, which is giving you 2 NADPH. And in the meanwhile, what is happening is, remember, 3 ATPs are generated. So 3 ADPs are used up to form 3 ATP. So photophosphorylation finally leads to the formation of oxygen molecule, 3 ATP, and 2 NADPH. Right? This is per 8 photons. And if there is 8 photons, how many electrons are there? That's for you to wonder, because that's a question on that, and we'll discuss this question there. Okay, that brings us quickly to the questions for today's session. Let's begin with this. Let's try to understand. So, if you remember in my previous session, we had left out some question on oxidative phosphorylation. So what we have done, or what I've done today here is, I have compiled the question of photosynthesis and previous session so that we get a revision of both the topics, right? Okay, right. So the first one is, which of the following statements are false? Chlorophyll B has a COCH3 group in place of CH3 group in chlorophyll A. The return from excited state to ground state during photosynthesis happens via electron transfer. The difference between the pH of stroma and thylakoid is around 3. 4 electrons are released when water is split by the oxygen evolving complex. It's basically the first complex. And Mg2 plus and Mn2 plus is central to the oxygen evolving complex. Whichever is false, do let me know. E is false. Yeah. Okay. So, uh, let's look at the first. Let's look at the true statement first. So, when there is a return from excited state to ground state during photosynthesis, that is via electron transfer. We have already established that. pH between the stroma and thylakoid Newman is 3. That's also correct. 4 electrons are released when water is split by oxygen evolving complex. That's also correct. Now, the fifth one, which talks about Mg2 plus or Mn2 plus being central to the oxygen evolving complex, that is incorrect, as correctly guessed, because the oxygen evolving complex contains manganese and calcium, but not magnesium as a central component. So it's mostly manganese that takes place. Right. Now, the first one, when you look into the structure of chlorophyll B, you will look as a on this C7, you will see that there is a CHO group. Now, you can also look into it as COCH3, but what I would say is, because when I am looking into the porphyrin ring, I have a CHO group. So that's why it's incorrect, because we are comparing the basic structure. So on the porphyrin ring, your chlorophyll A has CH3 and it has a CHO in case of your chlorophyll B. So that's why it's incorrect. Right. Next, now out of this oxygenic photosynthesis is basically used during your photosynthesis, oxygen is generated, right? And all the uh, organisms that do not evolve oxygen, those are called anoxygenic photosynthesis. Okay. Now here are some statements on oxygenic photosynthesis, and read the sentences, and then you can guess which are the true ones. One and three. Okay. One and three are true. Fourth, also, I just told about the homology. Well, let's come to it. So the first one, as you guessed it right, the plasma membrane of H. salinarum or Halobacterium salinarum. This does contain patches of light absorbing pigment, which are called bacteriorhodopsin, right? And this do not perform oxygenic photosynthesis, however. They use bacteriorhodopsin is basically used to generate a proton gradient for ATP synthesis, as we know, right? Now, the second one says ATP synthesis in bacteria, cyanobacteria, mitochondria, chloroplasts, all that we have just discussed do not share a common evolutionary precursor. Incorrect. We've just discovered that they are all homologous. They do share ancestry. Next one, many photosynthetic microorganisms obtain electron for photosynthesis from donors such as H2S and acetate. Now, here is the difference. This is what I was saying. So when you see plants, the electron donor there is the splitting of water. So you have water that is split into oxygen, and that is giving the electron for the plants or the chloroplast in plants. Now, for bacteria, they do not use or they do not evolve oxygen. They don't use water. Instead, they use organic compounds like H2S or acetate. So when they use chemical compounds, that's when it is an oxygenic photosynthesis. So that's what I was trying to say when I was saying that there will be a difference in the type of uh, electron donors that the systems or different organisms are using. Right. And as I've already mentioned that you do have different kind of photosynthetic. Photosynthetic term, photosynthetic means they are using chemical, they're using light energy to produce food or produce carbohydrates. Photosynthetic is basically synthesis using light, photosynthesis. That's true. But who are giving them the electrons? There is not water, rather that is H2S and acetate. So they are not oxygenic. They're anoxygenic photosynthetic microorganism. So if you just understand the terms, that makes it very simpler in life. Right. Fourth one says there is a functional homology between cyanobacterial cytochrome C6 and plant plastocyanin. So when you look into the overall machinery of photosynthesis or overall machinery of phosphorylation, you will see that there is a functional homology between cytochrome C6 and plastocyanin. Both of them are part of ETC or electron transport chain, and both of them transfer the electron to cytochrome B6 complex. We already know about cytochrome 6 that it transfers to cytochrome B6. Plastocyanin also does the same, right? So they both are doing the same function. That's why there is a functional homology. Okay. And the last one is modern purple bacteria have photosystem 1 and green sulfur bacteria have photosystem 2. And we have, while discussing only, we saw that green sulfur bacteria has type 1 and purple bacteria have type 2. So that's why it's incorrect. And you got that right. Okay. Next one. Now cytochrome B6F, which of these statements are true? A, B, C, and E. These are true statements. Okay. Yeah, correct. So A, C, and E. What did you say? A, C, and E. B is correct. Okay, cool. Right. So the first one says the cytochrome B6F complex contains a B-type cytochrome with two heme groups, a FES protein, and a cytochrome F, and that's correct. Statement 2, which is incorrect. Why? It says, the statement says cytochrome B6F conveys electron from a reduced quinone, which is correct because quinone is the electron acceptor there, quinone, to a water soluble protein that carries 2 electrons. Now, when you look into the photosystem or when you look into the path of electrons, you will see that cytochrome B6F transfer electron from quinol or plastoquinone to plastocyanin, okay, which is water soluble, is also correct. However, it is not 2 electron. It's a single electron carrier. It's a 1 electron carrier. And that's why that's incorrect. So the water soluble protein is plastocyanin, but it carries only 1 electron. That's why it's incorrect. Next is Q cycle. Q cycle, as we've already seen in the previous session, it's basically the cycling of electron. And we already saw this, right? That the Q cycle takes place in the BCF complex, which is basically when you have the uh, plastoquinol electrons is being transferred back to the ground state, right? Now coming to D. Now in chloroplasts, the direction of proton movement is from thylakoid lumen to stroma. When we did this here, in it we see it's not from lumen to stroma, rather from stroma to lumen. So it's actually the opposite, and that's why it's incorrect. And the last one says the measured difference in pH between the stroma and the thylakoid lumen represents 1,000-fold difference in proton concentration. That's correct. Right. Okay. Now, again, now this question is a bit of like it's from both the parts that we have studied. So, which of the statement regarding chemiosmosis and chemiosmotic theory is correct? A, B, D. Sorry. A, B, D is correct. C is also correct, right? That's right, correct? Remember, okay, I'll put it on the homology slide. Maybe that helps you. Here, all of these are F naught, maybe here. All of these are F naught or F4. That's correct, right? Um, well, the process is ATP hydrolysis, right? I mean, you call this as ATP hydrolysis only. And you also have ATP hydrolysis. I told you this in the last class also that there is also ATP hydrolysis that is taking place. Yes, yes, but it can also be hydrolyzed. How do I say? We also looked into how ATP synthetase can work in the opposite manner. I mean, I did not discuss it, but I told you in passing. Okay, no problem. Maybe in discussing the statements, we can see. So if you look into, I think it's there in the notes as well, where you have the other function of ATP synthetase, right? Look into this, there should be there. Or maybe I'll just check it up quickly. Anyways, so the first statement says the chemiosmosis is a combination of a transport process and a chemical process. Yes, we know that transport process moves your protons via the down the proton gradient. And the chemical reaction is ATP synthesis that we have already looked into. Second one says ATP synthase involved in proton transportation is basically two parts, FO and F1. Right. Third one says FO is responsible for proton movement and ATP hydrolysis can occur. That's what it's saying. So if you see, if it says can, which basically is so under some condition, like for example, low PMF, low protein motive force, you do have the hydrolysis as well instead of generation. So that's there. Okay. Oh yeah. And the fourth one says chemiosmotic theory states that energy derived from electron transfer reaction is temporarily stored as the transmembrane changes and charge and pH differences. Yes, that's correct. And last one says NADP reduction occurs at the end of the Kelvin cycle. So we will see that in the Kelvin cycle, it basically uses NADPH. So there is no more reduction there. The reduced component, which is NADPH, is being used up there. So it's not reducing any further. Yeah, did that point about ATP synthase may become clear? So when there is low protein motive force, ATP synthase can also function for ATP hydrolysis. Yeah, that's there. Remember this point. Okay, while I am looking into the notes if to check if this is present, this is a question in the meanwhile. So you have four reactions of the glycolytic pathway in this question. Which of these is not part of the gluconeogenesis pathway? And while you are solving this, I'll quickly check in the notes. Okay. Okay. Okay, sorry about that. So yeah, it seems like it's not mentioned in the notes, but yes, do remember that there is also this reverse function when under certain conditions for ATP synthase. Yeah. Well, coming back to this question. Okay. Uh, is the question visible to you? You have four enzymes here and four reactions. Which of these enzymes are not part of gluconeogenesis pathway? If you remember, it's basically I'm talking to you about the opposite. Yes, B and D, exactly, precisely that. But why B and D? I mean, precisely was I wanted to ask if that is being visible. B and D is E2 and E3, right? So they are part. Are you sure? I mean, you want to just think about it once again? Maybe I will give the explanation and then you can understand it better. Right, the first one is fructose 6 phosphate to fructose 1,6 bisphosphate. This enzyme is phosphofructokinase 1. And we know this is a highly regulated uh, step. And this is, remember, this is irreversible reaction. So uh, in the gluconeogenesis, this pathway is bypassed. It's not via simple reversible action of the phosphofructose kinase 1. You have a rerouting via which your this step of fructose 1,6 bisphosphate to fructose 6 bisphosphate takes place. So E1 doesn't take place in your gluconeogenesis. So that's why E. Now the second one, which is fructose 1,6 bisphosphate to DHAP and glyceraldehyde 3 phosphate that we, as you remember, is by aldolase, and this is a reversible reaction because they are constantly breaking and like basically these two are coming together, DHAP and glyceraldehyde 3 phosphate to form one fructose 1,6 bisphosphate. So this is part of gluconeogenesis. Third one, 3 phosphoglycerate to 2 phosphoglycerate is again a reversible step. It's towards the payoff stage. With the enzyme there is phosphoglycerate mutase, and that's also part of gluconeogenesis because this is also reversible. The fourth one, again, PEP or to pyruvate. The ultimate step is irreversible because that commits pyruvate into the glycolysis pathway. that is irreversible. To do this, again, there is a rerouting in gluconeogenesis via pep carboxylase and pep carboxykinase and pyruvate carboxylase. Remember, two enzymes in this step instead of pyruvate kinase. So that's why E4 is not part. So this was a kind of a refresher for you that uh, all these topics are concomitant, they're all together. So yeah, also we have this session 13. There are only 12 course lectures in this course, right? So that covers all the syllabus in this course. So lecture 12 will be the penultimate uh, session, where we'll be discussing the last course module or last module for this course. The section 13 is basically a revision for this course. So what I plan to do is I plan to discuss all the I will try to cover the questions from the assignments given this year. You already have the answer solution key with you. So we will discuss those questions, and in addition I will give you more questions. Instead of going over the topics, we'll solve questions, we'll do numericals, so that it's you get much more privy with the concepts and the solving, right? So this was kind of one of those questions you will get when we discuss in problem solving session 13. We'll have more of these revision questions where we'll do from first class to the last, and in no particular pattern. They will come in haphazard manner, right? This will aid you if you are taking exam. If not, it will help you understand the concept better, right? Okay, moving to the next question, right? Interesting questions. Now, intracellular concentration of ATP, ADP, and inorganic phosphate in 4 cell types are given below. So you have L, K, B, M. These are 4 cell types. You have their ATP concentration, and you have their ADP concentration, and you have the inorganic phosphate concentration. Now, out of this, which one of the cell type has the most negative delta G for ATP hydrolysis? You need to remember the formula. Try to remember the formula for ATP, ATP hydrolysis, delta G, and then try to understand which one of these 4 will have the most negative value. D. So C. And let's look into this how. Right. So first of all, I told you to try to remember the formula for delta G. We have already looked into this. Delta G is basically delta G naught, which is standard delta G, plus RT ln reactants, product by uh, reactant by product. So you have ATP hydrolysis. So your ATP is getting broken down into ADP and PI. So you have products by reactant, which is ADP, PI concentration by ATP. This is what you have, right? Now, you have several of these concentration, this and that. What you need to remember is for your delta G to be negative, this value to be negative, you would rather prefer a lower value of this. This value should be low so that this value also becomes low. Or rather, this value should be low so that this becomes also low, right? So, if your delta, in general, delta G as we have determined or we have seen is minus 30 kilojoule per mole, and the actual concentration is actual G is depending on cellular concentration, where you see that the equation favors when the equation shows a higher ATP concentration and a lower ADPD, this makes the ratio more negative. This is what you need to remember. If you have lower ADP and PI and higher ATP, you have a more negative delta G. So if you remember this concept, or if you remember this trick, or if you remember this idea, then it becomes very easy. What you need to do is you need to just calculate this part. Right? Everything else is anyway constant. So you calculate the ADPP by ATP. So you have 1.8 into 5 divided by 3 for L. For K, it is 1.3 into 3 divided by 3.9. For B, it is zero point 0.7 into 2.7 divided by 2.7. And for M, it is zero point 0.9 into 8 divided by 7.2. So if you do it, even by doing it mentally, we saw that this is zero point 0.7. Now, what I'm supposed to find out here is the lowest of ADP-PI. Because the lowest, the ADP by PI, my highest is the, or my delta G is more negative. Which one is the lowest? B is the lowest, and that's giving you more delta G. Now, I am expecting you did this. So let's say you got to the understanding that one is better. One is also there for K. So it should have been both the answers. But when you are looking by that logic, it's then when your C option becomes relevant or it makes sense. Did it make sense? Yeah. Okay. Right. Let's do this next one. So you have a standard free energy change for the conversion of PEP to pyruvate and ATP synthesis, which is given in the reaction that they take place. So PEP uh, plus water giving you pyruvate and inorganic phosphate, delta G is given. ADP hydrolysis, as we have just looked into, ATP rather synthesis is with the delta G 30.5. So the starting concentration of PEP, ADP, pyruvate, ATP, basically all the reactants and the product components are given to you. And the value of R is given to you. And you are to calculate the actual free energy change in kilojoule per mole for this reaction. We have done multiple of these types when we have to look at the half cell, and then we need to come to first the delta G difference, and then we use that to calculate the value of free energy change. So this is one more exercise for you for that type of question. The temperature here is 37 degrees centigrade. Take your time. We have done enough of these questions. I'm sure you will be able to do it. Yes, sure, sure, sure. Okay, so the answer is minus 27.8. Okay, let's look the left side first and then we will do it. Right. Okay, right. So mm, let's first remember this formula once again. We have delta G naught, delta G is equal to delta G naught, which is a standard delta free energy change, plus RT ln Q. Now, let's first find out this component here, right? We were given the free energy change, the standard free energy change for both of them, both the half reactions, right? Now, what is your reaction actually looking like? Your reaction is looking like PEP converting to pyruvate, that energy you have. ADP converting to ATP, that energy also you have. So delta G is basically both of these added together. So delta G naught for this entire reaction is minus 61.9 plus 30.5 is equal to minus 31.4. I don't remember if I told you last time or not. Maybe we can just do it once again. If you have reactions profile like this, if you are given the standard free energy change, you follow the direction and see which sign to use. Okay. And basically, if you have PEP to pyruvate is minus 61.9, which is what is here, and ADP to ATP is 30.5, you simply add them up because that's the way your standard free energy is given also. And that's what is given here. Your delta G is for the entire reaction is minus 61.9 plus 30.5, which is minus 31.4. You have the first component ready with you. R and T you already have. R is a universal gas constant, so that's given to you. T is in Kelvin. You need to convert 37 degrees centigrade to Kelvin, which will turn out to be 310. Now, what is this Q? Q, as we know, is the product by the reactant. So all the concentration, remember, this is what we just did here. So you need to do the same. All of these have the same unit, millimoles. So you first keep them in the same concentration, which is 50, 50 by 25, 25. That's what's happening given 4. Now you have delta G is basically minus 31.4 plus R T into ln4, which is basically here. Okay. So this is in joules. So we converted that into kilojoule per mole, and we are doing ln4, right? Now, when you solve this overall, you will see that if you substitute the value of ln4, which is 1.386 into this formula, you will see that this component to the right side of plus becomes 3.58, and when you finally add them to the delta G formula, you will get minus 27.8. Made sense? Okay. I think you still have some doubt. Maybe you can take your time because after this, okay, we have one more question left. So yeah, if you have any doubt, please post it out here. So these are the majority types of numericals that you will find in this course. So do practice this more. You will definitely find more questions on this. Okay, cool. So I will include this kind of question when we are doing the final revision, and you can have those revision there as well. Okay, cool. The last question here, which is also a part of the exam question or assignment questions previously. Which of the enzymes is responsible for arsenic toxicity? Which one do you think it is? Or have we done this already? I don't remember. But just in case, even if we have done or not, you can try to answer it once again. And then we can move on to the dark reactions part. This type of numericals are actually quite prevalent in the web. Like if you type out questions like standard free energy numericals, you will get most of these kind of questions. So you will find them under yeah, thermodynamics. You will find them under the initial set of topics that we read, right? So you can find those numericals there. Also, my suggestion would be you can try to solve gate papers, net papers. They often give this kind of questions, so you can have questions from there as well. So gate from the year 2014, net from the year 2014. Look into the biochemistry sections. You will get these kind of questions, and then of course you can type it out in the web. You will find more questions of these type. Yeah. Okay. Right. So, well, I'm so sorry. I didn't get the answer for this. So, yeah, this question is left. Then we'll do the next part for today's session. So, which of these enzymes is responsible for arsenic toxicity? I think I did. I don't remember. Yeah. So D, which is pyruvate dehydrogenase. And well, I think I did this. If not, then basically it's talking about arsenic toxicity, and you see the mostly in the pyruvate dehydrogenase complex, where you have the arsenic binding to the lipoic acid. But for the other enzymes, which is pyruvate kinase, aldolase, and phosphofructose kinase, they are not really affected by arsenic, right? Okay. Oh, there's one more. I'm so sorry. There's one more question. So the last question today is the Z scheme outlines the pathway of electron flow between the two photosystems and the energy relationship in the light reaction. What is the stoichiometry of photons for the overall following Z scheme equation? Basically, the overall reaction for Z scheme. And this is easy. I mean, this is what we discussed in the end. 8. Fantastic. Excellent. That's what. This is what we discussed, right? And we also have the values for each and every other component. Okay. All right. Now let's move on to carbon assimilation, which is the second part of photosynthesis. Now, what is carbon assimilation? So far, we have discussed about ATP generation, NADH generation, NADPH generation, this, that, oxygen evolution or oxygen evolving complex, which gives oxygen. Everything is there. Now, what actually is happening is we are, so these are photosynthetic compounds. They are synthesizing the food for themselves. What is the food there? It's carbohydrate. So that assimilation of the carbon into a carbohydrate is what is referred to as carbon assimilation reaction. This is has this entire process has several other names. Names like carbon dioxide fixation, carbon fixation, photosynthetic carbon reduction cycle, Kelvin cycle. All of these are basically referring to this one set of process reactions which belongs to or which overall leads to assimilation of the carbon dioxide. Now, this synthesis of carbon dioxide in animal cells, this uses precursors which have at least 3 carbon. That is what you need to remember. So not all the compounds can be used. Compounds that have at least 3 uh, carbons, they are the ones that serve as a precursor for synthesis of carbohydrate, right? And these 3 carbon compounds or these compounds which have 3 carbons, all of these are less oxidized than carbon in the CO2, which basically means if you look into the state of the carbon in the compound, they will be less oxidized than carbon dioxide, right? And then plants and other photosynthetic microorganisms. Uh, so this is, I was talking about animal cells. Okay, now in plants, what happens is that your carbohydrates, they are synthesized using the carbon dioxide and water. So you are basically here, what you're doing, because you're using water and you're using carbon dioxide, you're reducing the carbon dioxide at the expense of the energy used by ATP. And the reducing power furnished by the NADPH. So you're using this to do the opposite. You are basically using the carbon dioxide. You're reducing carbon dioxide and making carbohydrate. That is capable. That is what plants are capable of doing. And other photosynthetic, whatever is photosynthetic, whichever make their own food, they are able to catalyze this reaction of using carbon dioxide and water to give photosynthetic compounds or carbohydrates, right? So if you look into the image here on the slide, it shows you that the light driven synthesis of ATP and NADPH that we have so far discussed, these provide energy and the reducing power. When I say reducing power, I'm talking about NADPH, which is for the fixation of CO2 into trioses, from which all the carbon containing compound of the plants are synthesized. As we can see here, um, you have the sucrose, which is via for transport, which is, or you also have the starch for storage, you have the cellulose for cell wall. So these are the functionality of these different compounds, right? So all these come from this hexose phosphate. These hexose is basically 6 carbon, which come from 3 carbon triose phosphates. And these triose phosphates are coming from carbon dioxide and water via these currencies, which are generated by the light dependent photosynthesis. And these are all taking part in different plastids, right? So you have all these triose, which gives you hexose, which can give you all these different cell wall. Also, you have pentose phosphates, which becomes 5 carbon, which gives you the metabolic intermediate, which also gives you the DNA, RNA building blocks of cells, right? So you have every compound that is required for in a living organism coming from this fixation of carbon dioxide, right? Yeah. So we are going to now look into how all this is taking place. So as you can see, as carbon assimilation, if we are talking about, it's basically occurring in 3 stages. What are these 3 stages? First is the fixation. What is getting fixed? CO2 is getting fixed. And with what? What is the compound? The first compound that begins the entire assimilation process, it's the ribulose 1,5 bisphosphate. So a ribulose 1,5 bisphosphate with carbon dioxide gives you the first carbon compound, which is 3-phosphoglycerate. Next stage, this phosphoglycerate is reduced by the use of the energy currencies ATP and NADPH. So NADPH is being used and ATP is also being used. So it's an energy driven step. Your 3-phosphoglycerate is giving you glyceraldehyde 3-phosphate. And in the last step, you have to regenerate the ribulose 1,5 bisphosphate if you want to continue this step. So this same glyceraldehyde 3-phosphate via regeneration, it gives ribulose bisphosphate, again utilizes ATP. Now in this step, we will see further that not only is it giving rise to ribulose 1,5 bisphosphate, along with this, it is also utilized for glycolysis, because we know this compound is an intermediate for glycolysis, gluconeogenesis, sugar synthesis, starch synthesis. So you can see that one intermediate is being used for different different processes. So let's look into them in more detail. Oh, one more detail that I did not tell you was the number in the bracket is showing you the stoichiometry, the number of the ultimate number of compounds that is being used here. I will see it here in a while. So let's look into more detail. Stage 1 is fixation of carbon dioxide into a 3-phosphoglycerate. So your ribulose 1,5 bisphosphate is so with carbon dioxide is incorporated into this compound to give you the 3-phosphoglycerate. Now, who is doing this? A very important enzyme that you should definitely remember. The enzyme is ribulose 1,5 bisphosphate carboxylase and oxygenase, which is in short called Rubisco. So this Rubisco enzyme, this catalyzes the incorporation of this carbon dioxide into this ribulose 1,5 bisphosphate, which results in a 6-carbon intermediate. This is 5 carbon. This is 1 carbon extra, so 6 carbon. 6 carbon intermediate, which breaks down into 2 molecules of 3 phosphoglycerate. Right? So you have 2 molecules of 3 phosphoglycerate, and as I told you, it's a very important enzyme, Rubisco. So it is also a prime target for regulation, which we are not discussing. But when you are reading up, do keep a check on how is Rubisco regulated, right? So the first step gives you 3 phosphoglycerate. Now in the second step, now before I do go into that, this is like we mentioned here, right? Yeah, there is an intermediate of 6 carbon because both of these come together and then they break into 2 molecules of phosphoglycerate. This is how it is taking place. Now what is happening here is, if you look into the mechanisms of how is it actually taking place. So if you see the first step here, which is this. So you have this ribulose 1,5-bisphosphate. It forms uh, in a diolate at the active site. So basically it forms a intermediate. Now in that, after that intermediate is formed, you have CO2 which is in the proximity of the Md2 plus. This CO2 undergoes a nucleophilic attack by the indiolate, producing a branch 6 carbon sugar. So here you have the intermediate form, the 6 carbon compound is formed, which is also the beta keto acid intermediate. Then after that, you have the hydroxylation of the C3 of the sugar. So which is here, you have an H group being added, which is hydrated now. And now following hydration, you have the cleavage by aldol mechanism, which gives you 3 phosphoglycerate, 1 molecule. Right? Now this at first, when the 3 phosphoglycerate is formed, this leaves the compound, this leaves the complex. Then you have to have another complex, another 3 phosphoglycerate. So the remaining carbon ion, the carbon ironing of the remaining 3 carbon fragment is then protonated by the nearby side chain of lysine. Don't have to complicate. I'm just telling you how it is exactly taking place. So you have a lysine, it's protonating this carbon ion, and at the last, this phosphoglycerate is also released. So you have 2 phosphoglycerate not together, but one after the other. But what you need to remember at the end is that at this step, when you are fixing tribulose bisphosphate and you are fixing carbon dioxide, you are getting 2 phosphoglycerate. Now see, one ribulose bisphosphate is giving you 3 phosphoglycerate. So 3 ribulose 5-bisphosphate gives you 6 phosphoglycerate. That's how you need to remember. Now for 3 ribulose 1,5-bisphosphate, you need 3 carbon dioxide. So that is the stoichiometry that is shown here. Now let's look at the second step, which is the conversion of 3 phosphoglycerate to glyceraldehyde 3-phosphate. Now this conversion is again in 2 steps that are essentially the reversal of the corresponding step in the glycolysis. We have this step in glycolysis as well. Glyceraldehyde 3-phosphate to 3-phosphoglycerate. Here it's the same in the reverse order, right? So the 3-phosphoglycerate also gives an inorganic phosphate as a result. Now, the triose phosphate isomerase then converts these 3 glyceraldehyde 3-phosphate into, as we know, DHAP and glyceraldehyde 3-phosphate. After this, it becomes relatively simpler to understand. Now, 6 3-phosphoglycerate gives you 6 glyceraldehyde 3-phosphate and 6 inorganic phosphate. That's the stoichiometry here. And this is what is happening overall when you see, when you look into the processes, how they are taking place. This is what is taking place in the second stage. So you have the 3-phosphoglycerate here. It is converted to your glyceraldehyde 3-phosphate via this intermediate 1,3-bisphosphoglycerate. And then what happens is this glyceraldehyde 3-phosphate by triose isomerase and forms DHAP. Now this DHAP can shuttle out and move into the outside cytosols, and there it can be used to prepare or synthesize sucrose. While if it is in the stroma, it can be utilized to make starch or other compounds, as we will see. So your glyceraldehyde 3-phosphate and DHAP is what begins all the other reactions, right? And in the last, like I said, So this glyceraldehyde 3-phosphate, I told you, now some of them are moved out. This is what is happening. Some of them are moved out. One, basically. Now, what is the remaining doing? The remaining is basically giving back the ribulose 1,5-bisphosphate. And this step, when a glyceraldehyde 3-phosphate, which is a 3-carbon membered compound, becomes a ribulose 1,5-bisphosphate, as you can see, which is a 5-carbon member, we will see how it takes place. This takes place via these different steps. Not complicated if you try and begin and understand what's exactly happening. So if you look into this, it's basically showing the schematic of how there is interconversion between the triose phosphate and pentose phosphate. So if you keep following the black dots, it keeps giving you the number of carbons in each. So let's see. We begin by 3-carbon in glyceraldehyde, 3-carbon in DHAP. They two come together to form fructose 1,6-bisphosphate. Now this 1,6-bisphosphate gives out one phosphate, forms a fructose 6-phosphate. You have 6-carbon, 6-carbon still. Now this fructose 6-phosphate can give rise to erythrose 4-phosphate. Now, which enzymes are these? These are enzymes which are called trans-aldolase or trans-ketolase. Right? Now, your these third reactions basically is taking place by a trans-ketolase. So third, and you will see that subsequently when we come here, it is also a trans-ketolase, as you see here. There are several steps that are catalyzed by trans-aldolase. One, two. So you have trans-aldolase and trans-ketolase taking part in majority of the reaction. Anyways, so the fructose 6-phosphate converts via trans-ketolase into an erythrose. So from a 6-carbon, it has now become 4-carbon. This combines with DHAP, which is a 3-carbon, and 4 and 3 gives you the pseudohaptulose 1,7-bisphosphate, right? And this via several other conversion. Now we'll see how. So you have a bisphosphatase, one phosphate out. This one with ketolase, trans-ketolase gives you, so this 7 and this one here gives you ribose 5-phosphate. This and this together gives you one ribose 5-phosphate and xylulose 5-phosphate. This phosphate under isomerase gives you ribose 1,5-bisphosphate. So you have these several carbon numbered compounds 3, 4, 5, 6 coming together and giving rise to these different compounds. This looks very complicated, but it is not. It's just a logical flow. But yes, you need to remember certain things. For example, which steps are catalyzed by trans-aldolase, which steps are catalyzed by trans-ketolase, which is the final product that we are getting, what are the starting material that we are getting. This and what is the compound at the center, which is this pseudohaptulase. This is what you need to remember. Okay, and then this is final stoichiometric image here, which shows that you have 3-carbon dioxide with 3 ribulose giving you 6 phosphoglycerate, which gives you finally 6 glyceraldehyde 3-phosphate and DHAP. One of them shuttle out and go into the cytosol, while the remaining 5 gives you 3 ribulose by 5-phosphate, which gives you 3 ribulose 1,5-bisphosphate ready to be used again in this assimilation process. So you see very nicely regulated process is what you find here. Right? Now you see the stoichiometry here. This is a 3-carbon compound. So how many carbons in total? 5 into 3, 15 carbons. Ribulose is a 5-carbon compound. It is total 3. So 3 into 5, 15. So the number of carbon is still is just this aldolase and different processes that gives rise to your ribulose 5-phosphate. And we have just seen all of these are very much possible. It's basically moving from here to here, all these steps that we have just covered. You don't need all these intermediates, right? Okay. Right. Let's quickly wrap this up for today's class. So we have this question. Rubisco. So these questions are related to Rubisco. You can first try to attempt and then we will look into the answers. A, B, C. A, yes, C, yes, not B, but E. We'll see why. So the first statement is Rubisco is the most abundant protein in the biosphere. We have already commented on this. Second one says Rubisco exhibits octahedral coordination where Mg2 plus is coordinated with 6 hydrogen atoms. Well, that's not correct because in the Rubisco, Mg2 plus is coordinated with water, carbamylated lysine, the lysine that we saw here. This one. So carbamylated lysine and other residues, but not, it is not coordinated with 6 hydrogen atoms. It's with water. Okay. Then we have Rubisco has a relatively high optimal pH and its activation is regulated by Rubisco activase. Right. So I told you, you have to look into how Rubisco is regulated. So one of the important point that you need to remember is that it's basically activated by an enzyme for it to function, which is called the Rubisco activase. Next is Rubisco activase is an ATP independent enzyme. That's incorrect. Even Rubisco activase is an ATP dependent enzyme, right? It uses the ATP to remove the inhibitors from Rubisco's active site, when you read into the regulation, you will find it out, right? Okay, the next one says Rubisco function is carbohydrate biosynthesis. Yes, we haven't discussed this, so that's why probably, but it is part of all C3, C4, and SCAM plants, right? And last one says carbamylation of lysine 201 of Rubisco activates initiates the Calvin cycle. It is not Rubisco activase, it's Rubisco. That's the incorrect part. Okay, good job. All right. So next, moving to this. All right. So we have just discussed this. You have these three components, and you have these two enzymes, and you have these two products. So you need to match the following and tell me which one is correct and which one is wrong. Only A. That's it. Okay, I will wait when you are uh, done. Okay. Are you giving? Do you have any more answers? Okay, we'll do that. No problem. We'll do it together. Should be easy. So the first one you got it right. Next one is BSE. So this is what you need to remember. Yes, that's why I told you some key important reactions you need to remember. Let's look into this. First one says 3. 3 is xylulose 5-phosphate. Let's look at xylulose 5-phosphate. It's here, right? Xylulose for this thing. Then B, which is trans ketolase, which is correct. gives you 1, which is glyceraldehyde 3 phosphate. So it is correct. So that's why this is a correct statement or correct option. Next one says fructose 1,6 bisphosphate, which is here. It says A, so trans aldolase. This should give us DHAP. And it says third, so this is also correct. Now let's look into the other three. C says 2. 2 is pseudohaptulase 1,7 bisphosphate here, right? Then it says A. A is trans aldolase, so we can go here. Then it says 1. It should have erythrose 4 phosphate or DHAP, but it rather has glyceraldehyde 3 phosphate, so it's incorrect. Okay. Next one says 3, which is again xylulose 5 phosphate. So we can have it here or here, wherever you want to find it out. Xylulose bisphosphate with trans ketolase here, which gives rise to DHAP. Well, it is glyceraldehyde 3 phosphate here, and it is septohaptulase, which is incorrect. So this is also incorrect. And the last one says again pseudohaptulase 1,7 bisphosphate with trans ketolase, which you don't see reacting at all here. You have a 7 phosphate, which is with trans ketolase, but not a 1,7 bisphosphate, which gives rise to erythrophosphate. This reaction is by trans aldolase and not trans ketolase. So that's why it is incorrect. So did that, like, I understand why it would be confusing, but if you look into this schematic, it will become easier. But was it easier to follow with me right now? Cool. Thank you. Okay. Now, what are the stoichiometries for the following molecules involving carbon dioxide assimilation? So what you need to do is you need to first identify these three compounds. And once you do that, I am pretty sure you know which one is what. And anyway, we are talking about carbon dioxide assimilation. We have mainly three compounds here. Wait, not this. Let's look into this. This, and this. You need to identify these three compounds here, and then you need to tell me the stoichiometry for each. Three, six, six. Fantastic. Great. Right. The first one is ribulose 1,5 bisphosphate. Second one is 3-phosphoglycerate. And third is glyceraldehyde 3-phosphate. We know this one is 3. We know 3-phosphoglycerate is 6. And we know that glyceraldehyde 3-phosphate is also 6. So 366. Very good. Excellent. Yeah, the last question for today. So which of the following enzymes associated with Kelvin cycle requires light for their activation? So you have ribulose 5-phosphate kinase, fructose 1,6 bisphosphatase, pseudoheptulose 1,7 bisphosphatase, and glyceraldehyde 3-phosphate dehydrogenase. Which one of these enzymes require light activation? Confusing? Shall we look into the answer, or are you attempting? Okay. I'll give the answer so that we can. It's C. I'll show. I'll give you the answers. Okay, so answer is all of them, and that's because of this. So this you need to know that four of the Calvin cycle enzyme, which are all of these four, they are subject to special type of regulation by light, right? So what do they do? So all these four enzymes are activated by light-driven reduction of disulfide bond between two cysteine residues critical to their catalytic activities. So when these cysteine residues are disulfide bonded, which is basically oxidized, so these enzymes are inactive. So all these enzymes in their catalytic sites have two cysteine residues. If they are oxidized, they are not active. These enzymes are not active. Now, when there is a light, when there is light inhibition, electrons flow from the photosystem 1 to ferredoxin. Now, this ferredoxin is what helps them to gain activation. So your ferredoxin passes the electrons to a small molecule called thioredoxin. Uh, and this enzyme here is ferredoxin redoxin reductase. This reduced thyrodoxin donates the electron for the reduction of the disulfide bonds in the light activated enzyme. And this reductive cleavage activates the enzyme. Okay. So overall, if you look into this once again, sorry, just a second. Yeah. So you have light falling on the photosystem 1, which gives the electron to ferredoxin. Ferredoxin gives it to thioredoxin. Thioredoxin gets reduced, and the sulfur gets reduced. So thioredoxin, your sulfur get reduced, which it passes the electron to enzyme, and also in which also the cysteine S residues get reduced, and that is becoming active. So this thing, where is it actually taking place, we'll also see in next class when we look into the C3, C4. But yeah, the answer here is all the four via this regulation here, all via the thyrodoxin pathway. Yeah, right. So that's it. That's for today's session. So if you have any question, please type it out, or else we meet each other in the next session. Thank you so much. Thank you. Thank you for coming.